Okay, so today I'm going to finish up talking about blood clotting, and then we'll move on to a section students generally find review from organic chemistry because there's a lot of structural things about carbohydrates, so it's fairly straightforward material. Last time I got started talking about the importance of blood clotting and the importance of doing it rapidly, and I pointed out that the body can assemble a clot in the order of minutes from molecules, okay, and make a watertight bond that keeps you from bleeding to death. And I pointed out that that process happens as a product of a cascade of reactions. Now, the blood clotting process could be taught in an entire course. We're not going to focus on an entire course of material here. But what I'm going to do is focus on the bottom part of the process, because for our purposes, that's really where the nuts and bolts of blood clotting actually happen. Okay? Blood clotting can be initiated by a wound. It can be initiated by some damage to a vessel or something like that. So there's a variety of ways in which clotting itself can be initiated. We're not going to focus overly on that. But I will later talk about thinking about that wound itself. When we tear tissue, we actually rip open cells. And that turns out to be critical for part of the process I'll be talking about. Well, as I promised, we'll focus here on the bottom, and we're going to start our process thinking about the conversion of prothrombin to thrombin. So, all the things that you see here on this scheme are essentially proteins, and these proteins are, for the most part, enzymes. They're synthesized as zymogens, and then they get activated, and that activation results in an increasing signal of activation. Prothrombin is a zymogen. It is a precursor of thrombin, and when thrombin is active, it's a protease. Okay? So it's a protease. In fact, each of these guys are proteases because you can see one's activating the next, activating the next, activating the next. Thrombin is a protease, and its method of activation is that thrombin cleaves fibrinogen, and when it does it, it converts fibrinogen from an inactive form into a form that can polymerize. So the only one up here that's not a protease is fibrinogen, or fibrin. They're not proteases. Fibrin is a monomeric, protein. That is, I shouldn't say monomeric because it actually exists. It has three subunits. But it's the monomer of the clot. In other words, we assemble many of these together. I'm going to show you how that happens in a minute. Fibrinogen will not polymerize. Fibrin will polymerize. Big difference between the two. Okay? Fibrinogen will not polymerize. Fibrin will. All right. So when thrombin is present, it cleaves fibrinogen, and it makes fibrin. Now, one of the things we would like to have is we would like to have this process occurring at the site of the wound. We don't want this process occurring randomly, because if it occurs randomly, we might make a clot in our brain, might make a clot in our heart, we might make a clot in any place in our body and cause severe problems. So a key to this process occurring properly is that it occurs at the site of the wound, and prothrombin plays a role in this process that I'll explain in a little bit. Okay? All right. Now, let's look at the clot, the forming of the clot itself. Okay? This is the sort of schematic structure of fibrinogen. Fibrinogen has 3 polypeptide units. It's a dimer of 2 sets of 3. Okay? The 2 units are alpha, beta, and gamma. Okay? Alpha, beta, and gamma. The alphas sort of coil around. You can follow the alphas as a red. At one end, they have a structure we call capital A. And that capital A is essentially a block. It keeps the enzyme from forming a polymer. The beta similarly has a block. It's called a capital B at the other end. So when capital A and capital B are present, fibrinogen is just that. That is, it's fibrinogen when it has those 2 on there. If we take those guys off of there, then we will start to polymerize what results, which is fibrin, and make a clot. Okay? So if I take A and B off of alpha and beta, then this guy is going to naturally polymerize and start to make a clot. All right. So you'll notice that there are 3 proteins up here, alpha, beta, and gamma. Alpha has the capital A, beta has the capital B. It turns out that when these ends get removed, the portions that are left, okay, can actually fit into these grooves as you see here. And I'm going to show you a schematic of that in just a second. Okay? All right? Now, this shows a schematic of that polymerization. Notice here, if we focus on this little section right here, here were regions that had capital A's on them, but the capital A's have been removed. The capital A's, you'll notice, are fitting into the gamma holes, as it were. So the gamma has a receptor for that alpha unit once the A's have been removed. Gammas encapsulate the the alpha subunit. Okay? And there's an association between them which makes now a network of the clot. Okay? Now this network you can look at is only 2-dimensional. And that's because this gets really complicated when I try to draw it in the 3rd dimension. In the 3rd dimension, you're thinking about, well, what about the beta units? You cleaved off the capital B's too, right? The capital B's fit into, in this dimension, the holes of the beta chain. So one end of the beta chain fits into a hole at the other end of the beta chain and now makes a 3D meshwork tangle, if you want to think of it that way, that forms the basis of the clot. Okay? Now, this clot that you see right here, and even if I extend it out into 3 dimensions, is relatively weak. Now, why do you suppose it would be relatively weak? Any thoughts? It doesn't stay together real well. Well, it says you don't want to stay together forever. What did you say? No covalent bonds. There's only hydrogen bonds holding this guy together. Okay? So when we have an association, we haven't made any covalent bonds. All right? That becomes the job of an enzyme called a transglutaminase. And what transglutaminase will do is it will make covalent bonds between glutamine and lysine residues. So we can now imagine that wherever we've got glutamines and lysines in that mesh that are close to each other, a covalent bond is formed between them. We've just converted what I described originally as a loose clot, it's also called a soft clot, into a hard clot. Covalent bonds are not easy to break, and now this clot is going to hold against other forces that are causing it a problem. And if you've cut yourself, you probably have noticed that. You've probably seen that the very first clot that forms is very soft. And after it ages, it gets very hard. Okay? That transglutaminase is what's causing that to happen. It's strengthening the clot. Okay? That makes sense? Yes? A scab is a clot. That's exactly right. Yep. Okay. Now, that's kind of cool. I need to tell you something else. So, prothrombin turns out to play an important role in this process, not just because it makes thrombin, but obviously that's essential for the overall process. If it doesn't get converted to thrombin, then we don't make blood clots. Okay. There is a very interesting thing that happens to thrombin. I'm sorry, prothrombin. So I'm talking about prothrombin now, not thrombin. Prothrombin, the inactive form. All right? Prothrombin has glutamic acids, as most proteins have glutamic acids. However, prothrombin's glutamic acids get modified in a very interesting chemical modification. We've talked about chemical modifications of proteins before. Here is one you haven't seen before. This modification happens to prothrombin. It happens to a few other proteins in the body, but this one is very critical for the blood clotting process. What happens is the side chain of glutamic acids in prothrombin get an extra carboxyl group attached to them. So the side chains of glutamic acids in prothrombin get a side chain attached to them. All right? This reaction requires vitamin K. Requires vitamin K. Okay? Now, vitamin K, I forgot what I was going to say. Vitamin K does something. Vitamin K is an essential component of this reaction. If you don't have vitamin K, you don't put that modification on there. Well, why is that modification important? That modification is important because with that extra carboxyl group, it makes a perfect binding site for calcium ions. Those two carboxyl will bind to a calcium. Remember, calcium has a +2 charge, and these two carboxyl groups have a -2 charge, and they're placed in exactly the right space that the size of a calcium atom fits really nicely there. Okay? Now, why is that important? Remember, I told you that when you cut yourself, you cut open cells, right? We haven't talked about it yet, but one of the things I'll tell you later is that calcium is really a problem for cells. Cells keep calcium sequestered from the rest of the cell, and the reason that they do that is too much calcium will cause DNA to precipitate. So they have little stores of calcium that they kind of keep sequestered away from everything else. When you damage cells, you release that, or you at least open it up to now where calcium is available. You've just flagged where the site of the wound is. The presence of calcium at the site of the wound becomes a place for prothrombin to bind, and now prothrombin knows where the wound is. Prothrombin at the site of the wound gets activated, and as a result of that activation, which is all occurring in this same site of the wound, fibrin is formed and a blood clot itself is formed. Okay? That means then that if vitamin K is somehow inhibited, then we're not going to do that modification to prothrombin. We are not going to have the site of the wound, and we are in fact going to not form a clot. Okay? So that's a problem. Well, it's a problem. It's maybe not a problem. By the way, here's the calcium binding of the prothrombin, for what it's worth. Okay? Yep, I'm doing the wrong thing here. What I wanted to show you, here we go, okay, are some things that we call anticlotters. Anticlotters, okay? These are drugs that look enough like vitamin K that the enzyme grabs them, but guess what? They inhibit the enzyme. Now, this becomes very important if, for example, you're a person who tends to form blood clots. Maybe you've had a stroke. Maybe you've already formed an internal blood clot. Maybe you're more prone to this. Maybe you're getting ready, or maybe you've just had a procedure that might raise the risk of blood clots forming inside of you. And yes, that can happen. What you would like to do is reduce the likelihood that clots will form. So when that happens, people who have these likelihoods of happening are given anti-clotting medications, either coumarin or warfarin. They're actually related. And what they do is they work by inhibiting the enzyme that puts the carboxyls onto prothrombin. These are called blood-thinning drugs. Now, these you have to be careful with, okay? You have to be careful with them because clotting is important, folks. All right? If you're on blood clotting medication, I'm sure probably everybody here knows somebody who's on blood thinning medication, I should say, not blood clotting, blood thinning medication. If you know somebody on blood thinning medication, you probably know they have to go to the doctor fairly frequently and get sort of titrated to determine whether they have too much or too little of this in their system. And that means that the proper balance of these in the body, a certain amount of clotting you want to have, you don't want to have any more than that, these can affect that level of clotting that's happening in the body. Warfarin is also known because it's also the active ingredient in rat poison. And yes, it kills rats by stopping their clotting. Now, the first thing everybody thinks is, ew, I give this to rats and they just bleed to death in my house? Well, most likely not. Okay? Most likely what happens is one of the risks associated with too much or too little clotting is you hemorrhage internally. Okay? That's why the doctor is very careful with how much they give you, because you don't want, for example, your brain to form a burst blood vessel in some place and then you bleed to death in your brain. You know, and that's conceivable what can happen with anti-clotting medications. Okay, does that make sense? Clear as mud? Okay, so these guys are very important blood thinners. Sometimes people ask about aspirin. Aspirin, that's a blood thinner. I know somebody who takes aspirin to prevent problems associated with clotting. Is that the same thing? It's not. Okay, it's a very different mechanism. And again, aspirin involves prostaglandin, something you're going to hear more about next term. Okay? It's a different effect, and it's a different approach to slowing the clotting process. Okay. Let's see. So we've made clots, and clots are good for stopping things happening, but I suspect most of you, if you cut your arm, don't like walking around for 6 months with a scab, right? It's not, maybe it is, I don't know. Some circles it might be good, but in most circles it's not. Well, your body doesn't want to have that scab either. So just as the body has to have a way of making a clot, so too does the body have to have a way of removing that clot. After the tissue has started to heal, okay, getting rid of that clot is an important thing. And this involves another enzyme. Okay? The enzyme that dissolves the clot is called plasmin, Plasmin. It actually will physically take apart and digest that blood clot. Yes, it is a protease, but it has some other properties that are very important, including dealing with those covalent bonds that were there. Okay? Plasmin, we don't want plasmin floating around all the time because, again, if plasmin's floating around, it's going to simply destroy the clot as soon as it gets made. Don't want to do that. All right? So plasmin is made as a zymogen also. It's called plasminogen. 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 Plasminogen is the inactive precursor of plasmin, and it has to be activated before it becomes active plasmin. Why do I tell you this? I tell you this because this protease called tissue plasminogen activator, or TPA, this thing you see at the bottom of the screen here, is what converts plasminogen into plasmin. Tissue plasminogen activator converts plasminogen into plasmin. Now, this compound right here has great significance from the perspective of biotechnology history. Tissue plasminogen activator was the first genetically engineered human protein to be allowed to be used in human beings. It was produced by a company called Genentech, a very big company in biotechnology. And it was produced because it has some very magical effects in certain types of clots that form. Here's one. This is a heart that has suffered a blockage due not to cholesterol, but due to a blood clot. Okay? This blood clot was successfully treated with tissue plasminogen activator, which activated plasmin, which dissolved the clot and resulted in blood flow now arising as a result of that happening. Now, tissue plasminogen activator, or TPA, is not used widely for clots. There's dangers associated with it, as you might imagine. But in certain instances, TPA is used, like this, to save a life. In some cases, TPA is given to stroke victims. And if it's given in certain types of strokes within the first couple of hours, a lot of the effects of the stroke can be reversed. Because again, if the stroke arose as a result of a blood clot, then this can help relieve some of the problems that arise from the lack of blood supply, because it actually opens up the blood supply. Again, many different types of strokes, so we want to be careful in how we're using this. But this can be very effective in certain instances. Yes, ma'am?
So your body makes it to get rid of scabs and stuff, but why doesn't it make it for the blood clots?
So your body uses it to get rid of scabs and so forth. Why doesn't it make it for the blood clots? Well, it probably would. But think how long it takes to get rid of a scab. So it doesn't do a scab the minute it makes it. And when you're talking about a heart or you're talking about a brain, you're talking about something that's needed on the short term, on the order of minutes or hours. So that's the reason it's there. Eventually it would, but by that time you'd be dead, unfortunately. Okay? So tissue plasminogen activator was actually produced in a bacterium and was used in human beings. Since then, many other proteins, human proteins, have been engineered in bacteria and used in human beings very successfully. This guy is actually on the market and is frequently used. Okay. Questions about that? Yes? Yeah. Body does produce it, yes. That's what I'm saying. It's your body produces it to get rid of scabs. Right? So it's there to get rid of clots. But the body is making it over a longer period of time to deal with something that, as I said, that you would want to get rid of over a long period of time. But in the short period of time that you want it for a blockage due to in a stroke or something like that, having this to be able to give to a person is very, very beneficial. Do you see what I'm saying?
How would it detect the clot if you're injecting it?
How do you detect the clot? And I don't know exactly how it's administered. I believe it's actually injected near the site of the clot. I can't tell you that. I'm not a physician. I don't know that. Yes? What causes clots in people's legs and so forth over time after injury? There's not a single answer to that, but you could. But a lot of for example, there are some people who are very prone to clotting. There's a condition called phlebitis, for example, where people tend to form clots in their legs, and it probably has to do with that they're more sensitive to actually the banging type damage you're actually making in some cases. Clots to protect against that, so they're just more sensitive to it. But there's not one single answer to your question, unfortunately. Do you have a question?
The TPA, is it recognizing calcium?
Is TPA recognizing calcium? TPA actually is recognizing calcium to some extent as well. So there is a component of that that's involved in the clot. And I'm simplifying what I'm telling you. Yeah. Yes?
So is this kind of like a regulator? Like, does it click on kind of like how, what was the one we learned last week? Like, does this plasmin get like activated and deactivated then?
Does plasmin get activated and deactivated? Yes, it does get activated and deactivated. So plasmin will be taken out of action. Once you've made plasmin, that doesn't mean you're going to have it floating around all the time. So it does get deactivated, and I'm not talking about that here. Okay? Yes, one more question.
Can you go back over what the calcium does to prothrombin? It's not actually the calcium that converts prothrombin to calcium?
No. So the conversion of prothrombin to thrombin, his question was, can I talk about what calcium does with respect to prothrombin? So calcium is simply flagging the site of the wound and providing material for the prothrombin to bind to stay there. Okay? The conversion of prothrombin to thrombin happens as a result of action of another protease, which I haven't talked about. That's part of that cascade that I showed. Okay? Okay. So let's move on. We have carbohydrates ahead of us. Yay. Okay. How many people remember carbohydrates from organic chemistry? Yeah, right? Okay. Does anybody remember organic chemistry? Okay. Do you guys remember organic chemistry having a lot of structures and you memorize a lot of structures and that? Yeah. You're going to see a lot of that with carbohydrates. So I'm going to go over the memorization stuff kind of quickly because I think you've seen a lot of it before. But I will say it so that everybody has a chance to hear it if they haven't heard it before. So, carbohydrates. Carbohydrates they derive their name from the fact that they can all be written in the form of CX, where X is a number, parentheses H2O with an X. What does that mean? Well, if I have glucose, the structure of glucose is C6H12O6. Carbohydrate. Carbo referring to carbon, hydrate referring to water. So we think of carbohydrates as being hydrates of carbon. Now, there are some carbohydrates that violate that rule. But in general, simple carbohydrates will always be CXH2OX. Okay? Carbohydrates come in many forms and many sizes. And there are many names associated with them. Okay? So I'm going to go through and spend a little bit of time about that. The first thing I'll talk about is the fact that carbohydrates exist in two primary chemical forms, aldoses and ketoses. Aldoses are named for the fact that these are carbohydrates that have an aldehyde group in them. An aldose has an aldehyde group in it. One of the simplest aldoses is this guy right here, which is glyceraldehyde. You'll notice glyceraldehyde has 3 carbons. And you'll notice that there are, again, 2 isomeric forms. A form we call D and a form we call L. Okay? That's because the middle carbon has 4 different things attached to it, and those 4 different things can be arranged in 3-dimensional space in 2 different ways. You'll notice that the correspondingly simple ketose, which is simply a carbohydrate that has a ketone in it, doesn't have 2 forms. This is only true for the simplest one. And that's because the middle carbon only has 3 things attached to it. We will see numerous examples of ketoses that do have asymmetric carbons. This one's just too small to have one. Okay, so an aldose is a carbohydrate with an aldehyde. That's the thing up here in red. A ketose is a carbohydrate that has a ketone. That's the thing over here in red. Okay. We refer to these guys, you'll hear several names associated with carbohydrates. You'll hear sugars. Okay? That's because sugars are carbohydrates. They're a subset of carbohydrates. You hear saccharides. Saccharides you can think of as equivalent to sugars in some cases. Saccharide literally means sweet taste. Okay? A monosaccharide is a carbohydrate that has 1 sugar component. 1 sugar component. Okay? A disaccharide, therefore, will have 2 sugar components. A trisaccharide will have 3. A polysaccharide will have many. And we'll talk about those later. The nomenclature that we use for the monosaccharides is fairly straightforward. Okay? First of all, I've already talked about oses, aldose, ketose. You'll notice the OSE on the end. Whenever you see the letters OSE on the end, we're talking about a carbohydrate. So an aldose, therefore, is a carbohydrate that has an aldehyde. A ketose is a carbohydrate that has a ketone. Is this too much information? Is that... My brain is going to explode. Hopefully that's not going to happen. All right. Now, if I have a simple carbohydrate that has 3 carbons, we call it a triose. Well, now I'm starting to give you other terms. Is it a triose or is it an aldose? Well, it could be both. It could be an aldotriose, right? Meaning it has 3 carbons and it has an aldehyde. It could be an aldoketose. Will I count you wrong if you call it a ketoaldose? No, I won't. Okay? The most important thing is what the naming tells you, not that we get it exactly chemically right. I don't care about that. Okay? That is the naming. Tetros, 4 carbons. Pentose, 5 carbons. Hexose, 6 carbons. We'll see that the most common carbohydrates that we will talk about will be the pentoses and the hexoses. These are the most common carbohydrates that are present in nature, and they're the most common ones that we will talk about. Okay? Heptose has 7, octose has 8. We don't generally see monosaccharides bigger than 8. If they exist, they're pretty rare. Okay. Well, I told you that these guys can exist as different stereoisomeric forms, right? Well, let's look at some of these. Okay? This shows sort of a family tree, as it were. It's sort of a relational map. At the very top, we have the simplest of the aldoses, which is glyceraldehyde. Or the simplest one we consider here, anyway. Glyceraldehyde has 2 isomeric forms. That is, it has a single carbon that is asymmetric. 2 possible forms. I get 2 different forms of glyceraldehyde, L and D. All right? If I add another asymmetric carbon, I create a tetrose, and this is what a tetrose would look like. And this tetrose can exist in these different forms. Now, by the way, what this figure is showing you is all the combination of sugars that we refer to as D sugars. I'll tell you why we call them D in a second. There's another set of sugars that's not shown here of equal number that consists of the L sugars. You only see D-glyceraldehyde, you didn't see L-glyceraldehyde. Okay? Well, look at this. This guy has 2 asymmetric carbons. I see 2 forms on the screen. I could imagine there might be 4 different forms, 2 L's of each of those, because I'm only showing you the D's. So when I have 2 asymmetric carbons, I have 4 possible forms. I add another asymmetric carbon, I've got 3. You can do the math and figure out there are 8 possible forms. 4 in the D configuration and 4 in the L configuration. By the time I get up to 6, among the aldoses, I've got 4 asymmetric carbons. I now have 16 possible structures that I can have, different stereoisomeric structures of aldoses with 6 carbons. Okay? Are you going to memorize all these? No, you're not. Your faces are very glum, like Kevin's going to make us memorize all this crap. Okay? I'm not going to do that. All right? I'm only going to make you memorize some of this crap. All right? So what crap am I going to make you memorize? That's the question you're sitting here thinking, right? Okay? Get off the ball, Ahern. All right. Well, let me tell you, since that's already on your minds, because I've already brought it up, let me just tell you a little bit about which ones I think are important for memorization. And yes, I will expect you to memorize the structures of these. Okay? The ones I'm going to tell you, not all these. Okay? All right, here's our friend glucose, the most common sugar on the face of the earth. I think everybody should be able to draw this structure. Okay? Here's galactose, a related sugar. Okay? I think everybody should be able to draw the structure of galactose. Here's ribose. It may be every bit as abundant as glucose because it's found in RNA. Okay? Ribose is very simple. It's a pentose, 5 carbons. Look at this. All the hydroxyls on the same side. I just gave you an easy one. Okay? Now, there's one other monosaccharide that you'll need to know. It's a ketose called fructose. Those are the only structures of monosaccharides that you're going to need to memorize. Okay? Well, do we need to know both the D and the L? Well, if you know one, you know the other. Let me tell you how that works. Yes, question? I'm sorry? Sir, question, is this the form I want you to have them memorized in? I think you should know both the straight chain and the circular forms. And I'll show you the circular forms in a bit. Okay? Now, what was I saying? Fructose? D now. Oh, D now. Yeah. Okay. All right. So, what you see are D's. All right? The L form, please note this, is always the mirror image of the D form. It's the mirror image of the D form. So if I had, for example, L-ribose, and I said, draw me the structure of L-ribose, you would draw 5 carbons, just like this, and you would put every one of those hydroxyls on the left side. That would be the mirror image of D-ribose. That's pretty straightforward. If you had L-glucose, you would have this guy on the left, this hydroxyl on the right, this hydroxyl on the left, and this hydroxyl on the left. Okay? Now, I'm going to tell you something that's going to confuse you, so that's why I told you first the L is always the mirror image of the D. If I look at a sugar, and I don't know what the structure is, but I look at the sugar, can I tell if any given sugar is a D or an L? The answer is yes, I can, and I can do it very simply. All I have to do is look at the next to the bottom hydroxyl. If the next to the bottom hydroxyl is on the right, I have a D sugar. If the next to the the last hydroxyl on the bottom, next to the bottom carbon, not the next to the best hydroxyl, but the next to the bottom carbon, if the hydroxyl is on the right, it's a D. If the hydroxyl is on the left, it's an L. Okay? Now, the place where people make mistakes, and professors of biochemistry and Majors of biochemistry screw this up all the time. You guys are going to know more than them if you learn the following. D-glucose is not made by taking and putting that hydroxyl over there. What would you have to do? Mirror image. You got to flip everything, right? If you flip everything from D-glucose, you get L-glucose. Yes, you have to flip that one, but you have to flip everything else too. You can flip people off, and they'll be really happy in biochemistry. Okay? That was a joke. Okay, questions about this? There is. So his question is there a special term for when you just flip one of them? If you flip only one, you make what's called an epimer. Okay? If I flip any one of these four here, or any one of these three here, and I flip only one, I create an epimer. So what would be an example of an epimer? Xylose is an epimer of lixose. No, you don't have to memorize that. But if I gave you the structures, I'd say, how would you describe these two the relation between these two? They're epimers. The only place where they differ is right here. They only differ in one of those carbons. That's called an epimer, E-P-I-M-E-R. Okay? Now, a little bit more nomenclature. Okay, well, first of all, here's the ketoses. Same sort of thing. So ketoses can, in fact, have asymmetric carbons. There's the structure of fructose. That's one you're going to need to know. Okay? Below the second carbon, it's identical to glucose. Below the second carbon, it's identical to glucose. Okay. Now, enantiomer. I need to describe that word to you. Enantiomer is another word for a mirror image. D-glucose is an enantiomer of L-glucose. D-glucose is an epimer of D-galactose. If you want to go back and look at those structures and compare them, you'll see they're epimers of each other, meaning they only have one difference. Okay? A diastereomer refers to something that has differences in stereoisomers, but it's not a mirror image. Not a mirror image. Okay? If we look at this, for example, I could say that D-glucose and D-talose are diastereomers. They're not mirror images. They're not epimers, although diastereomers can be epimers as well. They're not mirror images. Enantiomer means mirror image. Diastereomer simply means different stereoisomeric configuration. Okay? So we have diastereomers, we have enantiomers, we have epimers. Yes? Okay, what features do they have to have in common to be diastereomers and not something else? They have to have the same number of carbons, and they have to be the same chemical form. So, for example, I couldn't compare an aldose to a ketose and say those were diastereomers. Glucose and fructose are not diastereomers of each other. Okay? How about we make it simple? I just told it to you in a complicated way. You want to make it simple? Diastereomers refer to anything that has changes in stereoisomeric configuration. We'll include enantiomers in that. We'll include epimers in that. That way you don't have to figure all these different categories. So diastereomer is the broad category of everybody. Includes enantiomers, includes epimers. Does that help? Now you don't have to draw these little divisions. Okay, so that's an important term. Now, let's go back here. As you probably remember from organic chemistry, sugars are long enough that you can twist bonds such that portions of them can be brought into close proximity to each other. And if you bring them into close proximity with each other, they will actually combine and make a bond. If I bring an aldehyde close to an alcohol, I'll make something called a hemiacetal. If I bring a ketone close to an alcohol, I will make a hemiketal. No, we're not going to memorize those structures. I'm not even that concerned about those names. The important thing is that we're forming a new bond, as a result of proximity, not unlike we saw with two cysteines coming together. Only in this case, it's occurring in the very same molecule. Okay? Now, when we do this with sugars, they can come together primarily in two different forms. They can form either 6-member rings or 5-member rings. If they form a 6-member ring that looks like this, we call them a pyranose. Pyrin is the name of this molecule, and we call sugars that come together in a 6-member ring pyranoses because they look like pyrin. If they form a 5-member ring, we call them furanoses because they look like this molecule, furan. And I'll show you an example in a second. Pyranose and furanose. Notice something here. How many carbons do you see in that ring? 5. You didn't see 6 carbons in there. You saw a 6-membered ring, but not 6 carbons in it. How many carbons do you see here? Keep those things in mind. Okay? Now, how does the ring structure form? The ring structure can actually happen in a couple of ways. And in this class, I'd like to keep it simple because you get plenty of structures to memorize as it is. We're going to consider glucose only when it makes pyranoses. And we're going to consider fructose only when it makes furanoses. And both of them can make pyranoses and furanoses. If you want to see how that happens, come see me. You probably did it in your organic chemistry and got sick of thinking of different possibilities. We're keeping it simple. We're going to consider glucose as pyranoses. This shows how glucose makes a pyranose. This happens when carbon number 1, there's the aldehyde, comes into close proximity with carbon number 5, remember 5 carbons. When that happens, what we see is that this bond comes in here, it attacks this carbon right here. That hydroxyl that was right there now is linked to that carbon. Okay? This carbon here that was a double bonded oxygen becomes now a single bonded hydroxyl group. And look what we just did. We just created a brand new asymmetric center. A brand new asymmetric center. Carbon 1 was not asymmetric to begin with, but after this cyclization occurred, this guy now has 4 different things attached to it. By the way, I'm not going to make you draw ring structures of L sugars. Okay? You're not going to draw ring structures of L glucose. You're not going to have to do that. Or L fructose, or L ribose, or L anything. All right? But because we have created a new asymmetric center, it can have 2 possible orientations. We call them up and down, or more commonly, up is referred to as the beta form, down is referred to as the alpha form. On the top you see alpha glucopyranose. It's in the D configuration. In the bottom we have beta D-glucopyranose. Okay? If I simply said alpha D-glucose, I would hope you know what I'm talking about. I'm talking about this guy. So there's a lot of different ways that people name these things. The pyranose part is not essential for name unless, of course, I ask you for that. Okay? Alpha D-glucose would refer to this guy. Beta D-glucose would refer to this guy. Okay. Now, this reaction to go from the linear to the circular is reversible. It's reversible. In water, it happens all the time. Now, why do I tell you that? Well, it turns out that if I have something in solution, for example, that's in the beta configuration, will it always stay in the beta configuration? The answer is, if it can go back to the linear form, it will not necessarily, because it can go back and forth and back and forth and back and forth. And it's doing that all the time. Yes, ma'am?
Is one more energetically favorable than the other?
Very good question. Her question is one more energetically favorable than the other? And it turns out that, yes, there is a form that's more preferred. The alpha form tends to be more energetically stable than the beta form. And we'll see a little bit why that happens probably on Friday. Okay? So in solution, we tend to see more in the alpha than we do in the beta. If you look at it, it sort of makes sense. The hydroxyl is down. It's out of the way from this group right over here, which is pretty big. Okay? Molecules are very aware of space and orientation. So staying away from that makes some sense. And because this beta configuration is the way that it is, we see some things happening with respect to structure that are even more interesting that, again, I'll talk about then. All right, so am I clear that this is a reversible process? Okay. Now, the key is that we have to be able to go back and forth. If we somehow stop this from going back and forth, we prevent the conversion from beta to alpha or alpha to beta. Well, how would I do that? Okay. A very simple way would be if I took that hydrogen off right there and I put a methyl group on there, it can no longer go back. It's frozen. So anytime I alter this hydroxyl, and by the way, this hydroxyl is on a special carbon. That carbon is called the anomeric carbon, anomeric. Anytime I alter the hydrogen on the anomeric carbon, I replace it with something else, I can no longer go back. You can't go back home, they say. Okay? Now, if I do that, if I somehow alter this hydrogen or this hydroxyl in any way, I create something we call a glycoside. So glycosides, underline this, cannot flip. They can't go back to the linear form. They're stuck in whatever configuration they happen to be in when they became a glycoside. Cannot flip glycosides. Okay. Here's fructose forming a fructofuranose. You see it's a 5-member ring. Well, what happened to those other carbons? Well, here's one. There's carbon number 1. There's carbon number 6. I always recommend when you work with a sugar, the first thing that you do, and nobody's going to listen to this, and everybody's going to get it wrong on the exam, like I said last time, always number your carbons. Always start there before you do anything else. Carbon number 1, 2, 3, 4, 5, 6. It will help you keep from getting confused. Last time on the exam, I said 50% of the people would miss, and they would try to tell me that Vmax over 2 is equal to Km. And what happened on the exam? At least half of the class told me that and got it wrong. It's wrong. Vmax over 2 is not Km. Now I'm warning you now. Number your carbons. You're going to have problems, okay? Numbering will keep you out of trouble, and you'll see why that happens later. Did I hear a question? Okay. Now, so, I look at this guy. This is in the alpha form. But Kevin, carbon number 1 is up. It's not carbon number 1 that determines it. It's the hydroxyl on the anomeric carbon that always determines it. This hydroxyl is down. We're set. Notice that the anomeric carbon is number 2, not number 1. Why? Because the double bonded oxygen was on number 2, not number 1. A ketose is always going to have carbon number 2 involved in the cyclization. Can't be number 1 because that would make it an aldehyde. Okay. Let's see. Just about done. I know it's a lot of material today. Let's get through. Here's ribose and deoxyribose. This is what ribose looks like in the ring structure. Okay. Again, it's very simple. This is beta D-ribose. Up. We got a total of 5 carbons. Number them. 1, 2, 3, 4, 5. Right? 2 and 3 are down. Very simple structure to memorize. The only difference between this guy and this guy is an oxygen right there. That's known as 2-deoxyribose, and that's the sugar, or the altered sugar, that we find in DNA, not in RNA. Okay? Okay, I think I'm running out of time, so that's probably a good stopping point. I will see you guys on Friday. You may leave this on? Shut it down. Yeah, I will, man. How you doing, Andy?
That thing work okay?
Yeah, it's been working fine. What I've found with this thing is if you turn the volume down a little bit, it actually works pretty darn well. So I've got it turned down where it's working pretty well right now. I had some real problems with it earlier in the term, so.
Everyone said they had problems.
Yeah, but once
I had problems with it this morning.
Did you? You did?
Yeah, it's
Oh.
feedback.
Well, when I came in, yeah, it was feedback again. When I came in, the volume was up, so I've got it turned to a volume now that's working pretty well. So you might try that. Okay? Take care. Hi. Excuse me. How you doing? Good.
Let me see what you said.